Perfect. So welcome everybody to the latest installment in our bee biogeography and systematics talks. This is hosted by the Center for Bee Ecology, Evolution and Conservation and the Packer Lab at York University. My name is Victoria McPhail. I'm the coordinator of what we call BC, and I'm joined today by the esteemed professor Lawrence Packer, as well as our guest speaker, Denis Michez. So to learn more about this series, visit yorku.ca/bees/Packer. I want to start off with land acknowledgement. We recognize that many Indigenous nations have long-standing relationships with the territories upon which York University campuses are located that precede the establishment of York University. York University acknowledges its presence on the traditional territory of many Indigenous nations. The area known as Tikaranto has been taken by the Anishinaabeg Nation, the Haudenosaunee Confederacy, the Huron-Wendat, and the Métis. It is now home to many Indigenous peoples. We acknowledge the current treaty holders, the Mississaugas of the Credit First Nation. We recognize that this territory is subject of the Dish with One Spoon Wampum Belt Covenant, an agreement to peacefully share and care for the Great Lakes region. I encourage you all to consider your relationship to the land that you're currently on, including the area that you also research or collect specimens from. If you aren't sure who is on your land currently or who historically has occupied and caretaken your land, visit www.native-land.ca, which gives you information for globally now about these Indigenous ancestors and current individuals or nations. And with that, I will turn it over to Lawrence Packer to introduce our speaker.
Hello, everybody. So it gives me great pleasure today to introduce Denis Michez, who is a professor of botany and systematics at the University of Mons in Belgium. And he's currently the head of the Laboratory of Zoology. His goal is to understand bee diversity and conservation globally. After completing a PhD in systematics, he's published species descriptions, phylogenies, and monographs. And he now leads projects on bee diversity evolution at population and upper taxonomic levels, including fossils, molecular phylogenetics, phylogeography, and bee-plant chemical interactions. I think he's probably got the largest research team of bee systematists and taxonomists working with him. Globally, that is. He's also developing focuses on global threats such as climate change, but still doing lots of taxonomy with researchers like Achit Dorchin working on Eucerini. He gave a talk in this series a little while ago. Thomas Wood working on Andrina. He's giving a talk in our series next year. Roman Le Divelec working on Hylaeus, Guillaume Gisbin and Pierre Asmont working on bumblebees. So there's a hell of a lot going on in Belgium these days, and we're just about to hear some of this. So over to you, Denis.
Thank you very much, Lawrence. I'm going to share my screen. Can you see it?
Yeah.
Okay, great. So thank you, Victoria, for the organization. Thank you, Lawrence, for the organization and the introduction. So today I'm going to talk about my first steps, actually, as a bee researcher. I worked on the melittid bees during my PhD. Since that, I did other things that I'm going to talk a bit during this presentation. But let's say that it's my first love is the melittid bees. So I'm going to start by, of course, presenting just a bit myself and the team that I'm working with now. So I work in the lab that was led by Pierre Asmont since last month, actually. So it just got retired three weeks ago. And so I'm the head of the lab now, and we have this nice project going on, including some project on taxonomy. So we were lucky enough to get some fundings to work on the taxonomy of European bees. And so I put here a slide with the faces of the PhD and postdoc of the lab and the technician. And you can see in the red frame, the one that they are working on taxonomy. There is Pierre, of course, who did work a lot on bumblebees and now is working on Anthophora. We have Simone working on Lagioglossum, Guillaume working on bumblebees, Tom that works on Andrena. And we have also Achik on Eucera and Romain on Hilaeus and also Maud on Nomada. So pretty good team of taxonomists, and we are trying to improve taxonomic resources mainly in Europe then. So to start my presentation on melittid bees, I have to talk about bee origin because I will talk about that right after. The melittid bees are now presented as the sister group of all the other bees. So the origin, it's pretty ancient. And so here you have a clade, very simplified, of the main bee family. Here the melittid bees are still presented as paraphyletic, but you have the main fossil, the main fossil that are important to understand this figure presented. You have the oldest fossil of sphaecid wasp, the oldest fossil of flower, and the oldest fossil of what is supposed to be a bee, so the Melittosphaecidae. You see that everything happened during the Cretaceous, and most of the bee family appeared probably during the Cretaceous, so before the Cretaceous tertiary boundary. And you see also the oldest fossil for each family, and there are quite a few fossil of bees. They don't preserve well, probably because most of the bees, they live in areas where the taxonomy is complicated. And so we don't have very old fossil of melittid bees, of any bees actually, from modern bees from Cretaceous, but you have a few from the tertiary. I will talk about that. So this is the basic hypothesis. And in terms of diversity and clades, we have two big clades, the long-tongued bees and the short-tongued bees, with more or less the same diversity. And in this diversity of bee families, the melittid bees is one of the smallest after the Strenoctrittidae that are endemic to Australia. So melittid bees have been always presented as a bit intermediate between the long-tongued bees and the short-tongued bees. They have a short-tongued bees, so they are not in the clade of the Megachilidae and the Epidae, so they are short-tongued bees. But when you look at the morphology of the tongue, it's pretty intermediate between long-tongued bees and short-tongued bees, because they have, so the segment of the palpalabia, that they are same length, but they have a mentum submention that it's close from the morphology of the long-tongued bees. So when Michener presented one of the first modern bee phylogeny, he presented the melittid bees as an intermediate family between the short-tongued bees and the long-tongued bees. So he presented the short-tongued bees as basal with the collected bees, because they had this apex of the tongue that is bifid, remembering the morphology of the wasp. And the other bees, they have a pointed glossa. So this was the hypothesis, the working hypothesis. And in this hypothesis, the melittid bees were intermediate between short-tongued bees and long-tongued bees. Some years after, there is this study of Alexander and Michener, 1995, where they analyzed more clades and consider more morphological data to present this phylogeny. And in these results, they present these two equally hypotheses, valuable hypotheses. one showing group melittid bees and long-tongued bees sister to all the other bees, and the other one confirming the hypothesis of Michener. And between these two hypotheses, actually, it's only with the new molecular phylogeny that have been developed by the lab of Brian Danforth that we were able to really stabilize the hypothesis about the phylogenetic relationship among the bees. And here on the left, you have this these results where you have as basal the melittid bees at this time. So it's a figure that comes from 2013. The monophyly of the melittid bees was not clear yet, but anyway, the melittid bees were basal compared to the other bees. And then you have a clade of long-tongued bees with the EPD and megachilidae. And then you have the other short-tongued bees. And this is the phylogeny that we are working with, I will say, since then. And the monophyly of the melittid bees have been confirmed in different studies. I cited here one, the one of Ranstetter 2017, so it's not that long time ago, using genomics. And so they show that the melittid bees are likely monophyletic, still basal and still sister to all the other bee families. So during my PhD that I started 2002, we started to work with Brian on a data set with molecular with sequences of some genes. And I published this paper that I will just show after, where the question was not about the monophyly of the melittid bees. We state that, okay, this is monophyletic, but about the numbers of subfamilies and number of tribes. So now I'm going to go deeper in the systematics of the melittid bees themselves. So there were different hypotheses on the table for the melittid bees. You have the one with, so the first figure A, the Michener 1981, where Michener state that melittid bees are monophyletic. And then you have some more, I would say, like a bit more fancy one, like the one of Engel, for example, 2001, where you have 4 subfamilies with the macropis being closed from the Dasypoda. So we tested this hypothesis. We analyzed 5 genes, and we also analyzed the morphology. We used the data from the study of Michener for the morphology, and we added some morphological traits on top of that, and the 5 genes. And so this is one of the main clades that you can see here that we find, where we actually confirmed the hypothesis of Michener, where you have 3 subfamilies of melittid bees. You have the one on the top, that is the Melittini, or Melitini, depending how you pronounce it. You have the Meganomiini, the red one in the middle, and then the green one at the bottom, it's the Dasypodaini. And in this first subfamily of Melittini, you have 2 tribes, the one of Macropis and the one of Melita, and you have 2 tribes in the Dasypodaini, the one with Hesperapis, Capicola, and Eremophanta, and another tribe with Dasypoda, Samba, and Aplomelita. So here, a figure that synthesizes a bit this phylogeny of the melittid bees. So this is the current state of knowledge for the family. So you have these 3 subfamilies. You have these 2 tribes in 2 subfamilies, and on the black bars you have the number of species. So you see that overall the melittid bees, like I said in the beginning, are not very species diverse. So one of the biggest genus here is Melita, and we are around 40 species. Hesperapis is very close, and maybe actually with a higher diversity, and then you have the Dasypoda. But you have some genus like Aphrodasipoda or the genus Promellita, where you have only one species. So overall, we are very far from the big groups like Andrena, Lazoglossum. Here, the melittid beast, I would say that it's a typical plesiomorphic group where you have a few species. Here is one of the map that I will show on the melittid bee. So here you have the distribution of the genera of the Dasypodaini. So you see that most of the diversity of the melittid bees are actually in the Old World. And for this subfamily, it's well exemplative. So you have the genus Dasypoda, for example, that is Palearctic. And then you have the Eremaphanta that they are Central Asia and they go to Oman. And then you have some genera that they are endemic in South Africa or Southern Africa, like here the genus Capicola. And you have some genera that they are in Northern America. Here you have the Hesperapis, for which the center of diversity is southwest of United States. So in one slide, but I will go deeper after on the ecology of the melittid bees. All the melittid bees are solitary, so there is no described species as social. Most of them, I would say like 95% of them are specialists in their floral choices. There is no kleptoparasitic clade in this group. All of them, they nest in the soil. And as far as we know, some of them at least do not cell lining. So for example, Dasypoda that is presented here, there is no cell line in the nest, but you have like a tripod on the pollen ball. You don't really see well this on the picture, but you have a tripod on the pollen ball to avoid the pollen ball to be directly in contact with the soil. And most of the species fly during summer. So during my PhD, I did this study at the family level, but I did also a lot of revision at the species level. And so here it's a short overview of the work that has been done at the species level. And when I'm talking here about the species level, the species concept that I explore is pretty basic. Here it's really based on morphology. Even if I did a bit of barcodes, I didn't have the time and the resources to do a very comprehensive revision, molecular revision of the different groups. But I have to say that based on the barcode data that we have now, the morphology is pretty useful for the melittid bees. What we can recognize as a species based on the morphology is confirmed by the barcode. We have a few cryptic groups in the Dasypoda probably, but we are working on. But overall, morphology it's quite useful for the melittid bees compared to other groups. So in short, we revised the Melita with Conal. We revised the Rediviva and Redivivoides. So it's a group of oil collecting bees from South Africa. We did the revision with Michael Kuhlman. The Macropis, the Promellita, and the Aphrodasipoda are done. Meganomia were revised by Mitchener. The Hesperapis, it's a big group still to be revised. So there is a full story about this genus. It was studied by Stage, the late Stage. So he died like 3 years ago, I think 4 years ago, during his PhD, and there are a lot of undescribed species with manuscript name, and so there is a work that is still alive. So I met Stage before he passed away, and I have his manuscript. We worked on together on this, and but the manuscript is so huge, like 150 pages, something like that, with a lot of description. And so to be short, it's in prep, and I hope that we will publish it not so long time. Capicola and Samba African genera are revised, Eremaphanta also, and Dasypoda is revised at the European level. Still have to work on the global scale. So what I choose to present here, it's not all the genera and all the work on the species level that I did or we did in the world, but I focus on the 3 genera that are recorded in Europe. And so there is Macropis, Melita, and Dasypoda. So now I'm going to talk a bit more about the species level. And so the species level for these 3 groups, the Macropis, the Melita, and the Dasypoda. So here you have a slide with the different diagnostic character for this genus Macropis. Is not that complicated to recognize. You have 2 submarginal cells. You have this scopa that is limited to the tibia and the basitarsus. And the Macropis, they are one of the few, I think, it may be the only one that it's oil collecting bee in the Palearctic. You have other bees foraging on oil in South America or in Africa or in Asia, but in Europe, Macropis is the only one foraging on oil. And so you have an illustration of this special hair that I present in the front leg and in the hind leg to collect the oil. And you have this structure that is a bit curved. And we guess that it's for capillarity, and so to suck the oil from the flower. Regarding the males, most of the species, they have a yellow face, and so they have this nice coloration that are the shapes are species specific, and then they have also this big leg, and then that justifies it's the etymology of the group Macropis, means big feet. So at the species level, for the 3 species that we find in Europe, it's pretty easy to recognize them based on the color of the face for the male here, but also from the genitalia. And overall, I'm pretty lucky with the melittid bees because the genitalia of the males are species specific. So it's pretty easy, at least for the male, to determine the species based on the genitalia. For the female, it's quite easy also. I don't have a picture for Macropis rivalski, but it's a very shiny cuticle, the same kind of shininess that you find in the Macropis in Northern America. And for the 2 remaining species, Europea and Fulvipes, the color of the scopa is different. So you have a black basitarsus for Europea, and you have a reddish yellow basitarsus for Fulvipes. So I don't have a nice molecular phylogeny to show for the Macropis, not yet, but I did this very rough phylogeny with the distribution where you can see the distribution that is all Arctic for this genus Macropis. You have 4 species in US, 3 species in Europe, and 9 species in Asia. But in Asia, you have 3 subgenera. So definitely the origin of the Macropis genus is Asia, and probably partly the tropical Asia. So I mentioned this when I described the diagnostic character of the genus. So the female is foraging on the oil produced by some flowers. In Europe is the genus Lysimachia, and I think in the rest of the world it's also that. So you have the flower and the female presented on the pictures there on the left, and the elaiophore that they produce the oil are at the base of the stamen, and so you have the bees foraging on the oil like that, and you have the pictures more a zoom of the hair on the right, and you still have some pollens of Lysimachia there. So during my PhD, I was lucky enough to have in my hands a fossil of Macropis sansulato. We describe a new genus, Eomacropis. This fossil is from the Eocene. It's from the Oise amber in France. This Oise amber is a bit older than the most famous Baltic amber. But so during this time, the Eocene, it was like 56 million years ago. And it's very impressive to see this amber with the bees inside, because you have all the hair, the color, the structure of the body that is still there. And you have a bee that is 56 million years old. It's really very impressive to study this fossil. Very well preserved. You see the wing venation is also very clear. And so we were lucky enough to see that actually there are some hair probably associated to oil collection there. During this time, the climate of Europe was tropical, subtropical, and so probably it's a kind of ecosystem that's where you find the same trees that they've been now, they are now visited by some group of oil collecting bees in Southern America. So probably the oil collection, it's very ancient. And so there was the hypothesis, we have two groups of melittid bees that they collect oil. We have the Rediviva in Southern Africa, and we have the Macropis in Europe, I mean, in Holarctic. So one of the hypotheses was that the Rediviva and the Macropis, they have a shared ancestor that were collecting oil. And what we find with the phylogeny is that it's probably not the case, because you have, as a sister group of Macropis, two genera that don't forage on oil. You have the genus Aphrodasipoda and Promelitta. And then you have the other clades of Rediviva and Melita that is sister to this tribe of Macropidini. So very likely the traits on oil collection appear independently in Macropis and Rediviva. So if you want to know more about the Macropis, there is this already old publication that we published, Seb Patini and me, in the Annal, where you have a key for the Macropis. So the second genus that I'm talking about today is the genus Melitta. So Melitta, they are less fancy than the Macropis. They're also, I think, a bit less pretty. They look like Andrena. So no offense to Tom, but to me, the Andrena looks a bit maybe less fancy in the color point of view, even if you have that very pretty Andrena. But overall, the Melitta are very brown, let's say that. And if you're not expert, I would say that you can misidentify a Melitta for an Andrena. They have some apomorphic traits. The scopa of the Melitta is limited to the tibia and the basitarsus, so you don't have this big flocculus that you can have with the Andrena. The shapes of the submarginal cells are not the same, especially the last one. The basal, the marginal cell is a bit, it doesn't touch the marginal vein. And you have, of course, only one submarginal, subantennal suture, while for the Andrena, you have two sutures below the antenna. And of course, you don't have this foeve on the side of the eye. So overall, these are the characteristics of the Melitta for the female and the male. You have some additional trait for the male that is illustrated there at the bottom left, is this last segment of the tarsa that is a bit enlarged, while it's more straight with the Andrena. So the identification of the Melitta are a bit more challenging, I will say, compared to the Dasypoda. They look more similar, but when you're in the field, it's pretty easy to recognize the species because all the Melita, they kind of have a different host plant. There are some species they have same host plant, but most of the time they are not sympatric, and you don't find a lot of Melita species living in the same community, I would say. So when you have the host plant, you kind of have the species. And here you have 5 among the most common Melita in Europe. You have Melita leporina on the left that is foraging on the Fabaceae, and here is foraging on the Melilotus. You have the up middle Melita nigricans that is foraging on Lythrum salicaria, an invasive plant in Northern America, but the bees is not there. And then you have Melita tricincta foraging on Onontites, Melita dimidiata foraging on Onobrichis. So Onobrichis is a genus of Fabaceae. And then you have Melita hemorrhoidalis that is foraging on Campanula. So pretty different host plants. Here you have the map of the Melita genus. You see that the hotspot of diversity is in Europe, and especially the Mediterranean basin, like most of the bee genera in Europe. The highest diversity are always Spain or Turkey, Greece or Italy. And you have some species in the East Palearctic, going to Japan. You have 3 species in US. Some of them are foraging on Ericaceae, if I remember well, on Vaccinium. And then you have a few species in South Africa. So as most of the diversity is in Europe, what we expected when we did the molecular phylogeny of the Melita with Simon, it's to find the European species basal, but it was not the case at all. So when you see these clades, and I have an illustration here with the distribution mapped on the clades on the left. You see that actually the basal species, so Melita Richtervandensis, Melita aventurensis, or Melita schultzei, are actually basal in the clades. And so we have for the Melita a scenario out of Africa. So the plesiomorphic, the basal species are African, and then you have colonization from Africa to the Palearctic, and then from the Palearctic, the Nearctic. You have also on the right the host plant choices mapped on the clades, and you see that you have a lot of change, but you can also see some irritants during the speciation process. So, for example, you have in green beige the clade of Hemorrhoidalis tomentosa melanurabudensis. All the species forage on the Campanula. So you have host plant shift that is quite wide in the Melita compared to other groups. You have shifts from Fabaceae to Scrophulariaceae to Campanulaceae to Fabaceae, but when inside one clade, you can have some stable host plant choices. And for example, up, you have Equorti and Melitoides. They are both specialists on Ericaceae, the 2 species that they are in Northern America, while actually any other Melita in the world are foraging on the Ericaceae. So with the Melita, we went even a bit deeper than the species phylogeny. We did some phylogeography, because we were wondering what is the impact of these floral choices on the genetic diversity and the genetic structures of this of sister species. So we thought that we had a very nice model to really analyze what is the impact of the distribution of the host plants on the genetic diversity and structure of the bees itself. So you have these 3 species, Leporina nigricans tricincta, that are already introduced before. And so Leporina is foraging on a very common host plant, the Fabaceae, that you can find almost everywhere. So our hypothesis was, okay, the size of the population of Leporina can be bigger, and also the connectivity of the population can be bigger, so the structure, genetic structure, will be lower for the Melita leporina compared to the other species, nigricans and Tricincta, where you have localized host plants or very rare host plants. So we sample these 3 species through Europe, and even for Leporina, we went to the Baikal Lake. It was an amazing trip there. So you can see the dots on the maps on the left. The 2 maps on the top are the maps of the population that we sampled for Leporina, and the 2 maps in the bottom left, it's nigricans, and right is Tricincta. And then you have on the right the result from the haplotype analysis, and you see that indeed the haplotype diversity of Leporina is pretty big, and the structure of Tricincta, which is foraging on the most localized host plant, the more fragmented host plant, has a stronger structure, even if the diversity is lower. And nigricans is pretty not structured, actually. And the fact that nigricans is not structured, it's probably because this species is probably the youngest and colonized Europe pretty recently. And so here you have a couple of heat maps showing population diversity and individual distances. But I will not go through all the details of these maps, but in a few words, if you look at the 3 maps on the top, on the left, you have Leporina, on the right, you have Tricincta, and in the middle, you have nigricans, and you see that the colors are much more red for Leporina, meaning that the genetic diversity is much higher than the nigricans and Tricincta. If you want more information about the Melita, you have these 2 papers that I present here. And I will finish this talk by one of my favorite genus of bees is the Dasypoda, so the Patalon bees. And here again, I show you some diagnostic characters to recognize this genus. So you have 2 submarginal cells, like the macropis, but the intervein between the submarginal cells is straight. So do a straight angle, and you have this huge scopa of the female. So Dasypoda means hairy leg, and indeed they have very hairy leg. But again, the scopa is limited to the tibia and the basitarsus. You have on the males also quite this trait also of long hair on the basitarsus. So here are some pictures that we did with Vladimir Radchenko from Ukraine. So just a thought about our colleague Vladimir, who is really struggling there in Kiev, but he's still working on the bees and he's still sending us pictures in the framework of European projects. So he's doing this kind of amazing pictures. Here you have the species Bracata. So you see this bee that really, I think that we can brand this kind of bees with very nice color and very hairy leg. Here you have the structure of the tongue. So you see that even if the melittid bees are short-tongued bees, the tongue can be pretty long, actually. You have variation in this group of the punctuation of the clypeus. You have also variation in the malar space, the length of the malar space, variation in the pilosity, of course, variation in the length of the galea and the relative length of the palpa and the galea, and the sculpture also of the galea. So some closely related species, they can show variation in this in the sculpture of the galea. The antenna can be also useful to recognize the species, the pilosity of the metasoma, And of course, like I said, for the melittid bees, the genitalia and the hidden sterna, so sternum 6, 7, and 8 are pretty useful, and it's quite easy when you see these pictures. You have different species, and it's pretty easy to recognize the different species based on this. And here, the genitalia of the Dasypoda. So you have the gonostyli that are more or less divided, and they can, in this different part of the gonostylus, can be more or less joined. And so here you have some examples of, on the left, of a genitalia of Dasypoda sensu stricto, where you have two structures of the gonostyli, but you have a membrane joining the two. In the middle, you have just one gonostyli, and then on the right, you have three structures on the gonostyli. So the distribution of the pantalud bees are this one. So it's Palearctic, but we find recently two populations of a new species that is named Dasypoda riftensis in Ethiopia and Tanzania. So I don't know if Lawrence collected some Dasypoda in Tanzania, but it sounds like a very interesting places where you still have some Palearctic genus like Dasypoda. They are there. So during my PhD, I did a rough phylogeny based on the morphology of the Dasypoda. And so here the presentation of this phylogeny with the mapping on the character mapping of the host plant choices. And what is cool with the Dasypoda is that we have a match between the pherite choices and the systematics. So basically the mega Dasypoda that I will present right after, they forage on Dipsacaceae, no Caprifoliaceae. The Dasypoda sensu stricto, they forage on Asteraceae. And then the two other genera, Heterodasypoda and Microdasypoda, they forage mainly on Cystaceae. And what is also interesting is that the morphology of the setae of the scopa are look adapted to the host plant. So you have different morphology of the setae of the scopa for the Dasypoda sensu stricto or the Megadasypoda. But so overall, I described 4 subgenera based on the morphology. And I will introduce briefly the characteristic of this subgenera. So you have the mega Dasypoda, and so like they are named because of that, they are the big Dasypoda. They are big species, big meaning like 16, 17 millimeters length. They have a long malar space. They have this galea that is really long compared to the maxillary palpae. And they have also this pygidial plate that is hairy. Don't know why. I don't know if there is a function to this, but they are the only group with hairy pygidial plates that you can see on the right bottom. Here you have some traits of the males, and the genitalia are pretty distinctive. You have these 3 lobes of the gonostyli. You have also the apex of the 8th sterna that it has 2 teeth, and also the sterna 8 that it has short tooth. Sterna 7, sorry. Here you have the Dasypoda sensu stricto, that they have a shorter galea, and there is a hairless pygidial plate, shorter malar space also. And the males, they have a very distinct genitalia with hairs on the gonostyli, and they have also, you can see bottom right, this hook at the base of the 8th sterna. That is very characteristic of this group. The other 2 subgenera, the Microdasypoda and the Heterodasypoda, they are smaller and they are pretty similar in the morphological point of view, but the 2 subgenera, they show a short galea compared to the maxillary palpae. And they show also a bicolor scopa. So you have dark hair and reddish hair. You have variation in the clipeus punctation and variation in the genitalia and the sterna. So I will go fast on this. The cool thing also with the subgenera is that the biogeography fits match also the phylogeny and the host plant choices. So the center of diversity of the Megadasypoda is clearly the Balkan and Greece. So in Greece, you can have up to 6 species. That's what is presented on the left. And for the Microdasypoda, the center of diversity is clearly the west of the Mediterranean basin. So, like I said, we are working on the collection of new molecular data, barcodes, but also this UCE that we are producing with Brandstetter. And the cool thing again is that what we find with the morphology is confirmed by the UCE. So the different groups and the different subgenera that we propose based on the morphology are the same, basically. So you have on the left here the Dasypoda sensu stricto, in the middle Megadasypoda, and on the right hetero and Microdasypoda. What is also interesting here in this phylogeny, I put an arrow there to show the phylogenetic position of the species that we find in Ethiopia and Namibia. No, not Namibia. Yeah, in the east. This is not a basal position like we have in the Melita. So in the Melita, we had a few African species, but they were all basal in the genus. And here, the origin of the Dasypoda is clearly Europe and the Mediterranean basin. And you have a movement from Europe to Africa. So if you want to know more about Dasypoda, I'm afraid that now you have to learn French, because these are my first paper that I wrote when I was a PhD student, and I was advised to write it in French by Pierre, that I stopped after, fortunately. But we are working on the revision of this group at world level, and so this publication will be in English. So a few slides to finish my talk on the perspective. So on the melittid bees, like I said, we still have to work on the Hesperapis. It's the last genus that doesn't have a revision for the family. And also we are working on a paper in English at the world level for the Dasypoda. And this is a part of what I'm doing in the study of bees. From systematic, I moved after my PhD, I moved to conservation biology, ecology, evolutionary biology. And so I'm convinced that taxonomy is the base of natural sciences. And so I use my expertise as taxonomist to feed with good evidences conservation plan. And so in Europe, pollinators are quite top in the agenda in conservation biology. And we have this program that we call the Green Deal, implemented by European Commission, and they are trying to develop first action plan, and then implement this plan. And so I was lucky to participate to a species action plan for the melittid bees, and we decided to focus on the Dasypoda. They are specialists on the Caprifoliaceae. So the one that is illustrated here on the left, Dasypoda suripes, because these species are declining. They lost half of this distribution, if not more, because these meadows with Caprifoliaceae are really threatened with nitrogen, in Northern Europe, habitat destruction, so many threats. It's not the topic of this talk, but again, we are using the information that we have from all this revision, the maps that we produce, the keys, to implement monitoring and to implement a conservation action plan. And on the right, the 2 books that we published in the lab recently, Bees of Europe and Bumblebees of Europe. And our goal is also to produce more of this kind of books or studies to have in the future. This is the the big perspective, but I hope that it's possible to have a key at species level for all the European bees. So it means 2,100 species in the next decade or in the next 2 decades to be able to have this ambitious monitoring at species level. So I'm stopping here. Thank you all for your attention. And if you have any question, I would be glad to answer.
Well, that was a lot of fun. Thank you very much. Great pictures, beautiful bees.
Thank you.
Some interesting parts of the world you've been to. Lake Baikal. Wow.
That's fun.
So, a question from Anna Carolina Isapan Mota. Does the literature mention something, or do you know what were the selective pressures on species of Melitta occurring in South Africa?
The what? The
selective pressures
to go out of Africa or for the fried choices?
Yeah, I'm just reading the question.
Okay.
And can you add more to your question, Anna, so it's a bit more precise?
So if I can start to answer to this, so when you see the Melitta, definitely what they like is temperate Mediterranean climate. So they don't like tropical. They are not there. Any melittid bees actually are there or subtropical. So I guess that they were able to move out of Africa by the east when you had this contraction of the tropical forests, and they reach Europe and the Palearctic through this way. And so this is kind of movement that you see in a lot of groups that they are present in Southern Africa and Northern Africa. I'm thinking about the Fidelia, for example, where you have also quite an old group that you have one species in Morocco and the rest of species in South Africa. And so to move, for sure, one of the constraints was to have some paths that are not too wet and not too tropical. And this is the kind of things also path that you see maybe in this Rift Valley, in the mountain also, in some area of Tanzania, of Kenya, of Ethiopia. And so you see this movement still now, or not the movement, but let's see, you see trace of this movement in the east, in the Rift Valley. And this part of Africa, it's probably super interesting to sample in the future, because you have really this movement between the Palearctic fauna and the sub-Saharan fauna. And there are probably a lot of things to discover there in terms of species or group. So I guess that the main
Sorry, yeah, you're dead right. Eastern Africa is going to have a lot of interesting things there. And Alain Pauly is living most of the time in Tanzania right now, right?
Yeah, that's him who find this new population of Riftensis in Tanzania. I knew that you will talk about that, so I have the data. Actually, so he collected the Dasypoda in Kilimanjaro region, Machame Road, on an Asteraceae, on the Lonaea cornuta. That is kind of the same plant that where you find the Dasypoda in Ethiopia, in the south of Addis Ababa.
So it's almost certainly in Kenya as well. It's just that nobody's caught it there yet. I've not been to Tanzania. I have been to Kenya though.
Yeah.
So yeah, it's fabulous stuff. So when was it that that area was more arid?
That's a good question also. I think that it's quite I mean the movement of the tropical forest, it's a lot, it's many events. I mean, it's related to a variation that it's shorter than the geological age. You know, the Sahara not long time ago, it was green. So
Right.
this area is, the constriction is really frequent, I would say. So I don't know, we didn't do a molecular clock on this. I don't know when they passed, but what can I say is that there are probably many occasions. It's not just one, just not a land bridge. It's really something that is variating a lot there.
But you are talking a lot older than the glaciations, right?
Yes, probably it's older than glaciation, yes. But I don't know. There are a lot of things that still happen in the Miocene, I think. Huh? But I don't know. I don't know. I don't want to guess based on things that I don't know.
Right, but you'll get the data soon.
I hope
So,
so, yeah.
so a question from Eugene Scarpula. Have you ever noticed variation in the number of submarginal cells within individual species or on individual specimens? In other words, asymmetry.
No, I didn't see that. I saw that on Andrena, for example, but I never saw a variation in this point of view. It's really, it looks stable. So we have these two submarginal cells on Macropis and Dasypoda in Europe, and the Capicola and the Remaphenta and the Hesperapis. And we have three submarginal cells in the Melita and the Rediviva. And it's pretty stable inside each genus. It's stable. There is no variation inside the genus.
Yes, some years ago, Eugene published a review of submarginal cell number variation in bees, and it crops up all over the place, but apparently not in melittids.
No.
An anonymous attendee asks, what do you think is driving the pattern of host plant choice in Melita? Do you know if these plants have common traits?
Yeah, that's a good question. There is a paper that I didn't talk about. So what we wonder why the Dasypoda, when they shift host plant, they look to go on similar clades or at least similar morphology. While actually, when you see the shift of the Melita, they go from Fabaceae to Campanulaceae to Lythraceae, so very unrelated plants. And so the hypothesis was, so it's not the morphology, because Fabaceae has a bilateral symmetry and Lythrum has a radiate symmetry, for example. So it's not the morphology of the flower. So the hypothesis was the chemical composition of the pollen or the scent, the perfume of the flower. And we find some similarities between the chemical composition of the plants, but not necessarily related in the sense of when you compare to the other plants, they don't make cluster. So they have similar chemical composition, but there is no one chemical compound that was common to the host plant of the Melita that could explain that they can shift from one to the other because they have similar composition. So it was not the case. We find this for the Colites, but we didn't find this for the Melita. So how they do it? One of the hypotheses that we were not able to explore is that the Melita, they have a big Dufeu gland, and maybe one the thing that they could do is to add some chemicals into the pollen loads that makes them able to really feed on very different pollen. And then, so they will be kind of free of the constraint of the chemical composition of the host plant. While, for example, for the Dasypoda, what we find is that they tend to go on plants that they have cholesterol, so a lipid that they use to make the molting hormone. So maybe it is that the thing is that with the Melita, to test this hypothesis, we have to find a nest, and we have to find the pollen ball, which is very complicated for the Melita. So the chemical composition is not a constraint, the morphology is not a constraint. So why they are shifting? I guess that they go on plants that are quite abundant. And when you see the Lythrum, they can be very abundant. They are localized, but when you find Lythrum, you can find a lot of Lythrum. So I guess it's that. And the same for Odontites or for Fabaceae.
That's fabulous. So there's plant chemistry involved here and adding things to the pollen ball in a group that's related to these oil bees. There's something going on there.
Exactly. And I think also that with the for the melittid bees, you know, as they are basal, the problem is to find them. It's complicated to find the nest, but I mean, we can maybe find with the melittid bees some answer about how you have the shift from the carnivorous diet to the pollinivorous diet, you know, how the primitive bee was able to feed on plants instead of feeding on prey, you know. And what is interesting with the chemical analysis that we are doing with the Dasypoda is that, so for the sterol, so the insect, they don't synthesize sterol like the vertebrates. So they have to find the sterols in the food, and the sterols are the base of the physiological pathway for molting hormones. So if insects don't have sterols, they don't molt. And what is very interesting is that the Dasypoda are growing on plants that produce cholesterol, like the cholesterol that animals produce. So the one that you find in the prey of the wasp. While most of the plants, they produce phytosterols, that are sterols with additional carbons. And so you can think that this protobee that shifted from one diet to the other one could maybe shift on this plant diet because the chemical composition of the pollen was similar to the chemical composition of a prey. Talking about proteins and talking about sterols, for example.
That's absolutely marvelous. Have you published that?
Not yet. We don't have enough data for that. So it's just a hypothesis.
Well, it's a great idea. That's marvelous. Dino, it's great to hear from you. Dino Martins, who lives in Kenya. Clearly so much more to do in our part of the world. Do you think more sampling in Africa will reveal different patterns in terms of biogeography and phylogeny?
Yes. I think that it's one of the most interesting areas to sample with Central Asia. This Gobi Desert and Central Asia is very interesting, and all this Rift Valley is also super interesting. And we need more people working there for sure. And so in the lab, so Achik Dorshin, who did a talk on the Eucerine, actually now he's going to work on the African bees. So we have Alain that is there, and Nico, so Verreken is working on the African bees, and we need definitely more African working on African bees. So in Belgium, we have the chance to have Tervuren Museum, which is the Royal Central Africa, because of colonies. And so definitely we want to do more work there in Africa, and especially in this Central Africa and Eastern Africa.
Yeah, very, very interesting. Yeah, there's going to be tons of neat things to find there. You know, there's still, what, 2 Meganomiine species known from single specimens from Madagascar. I think, what, they're subgenera known from single specimens. So yes, Madagascar is another place.
And there is one specimen from Yemen also.
Oh, right. Yeah.
And so it's a South African group. There is also in Madagascar. They are also in Nigeria. But I'm sure that they are there in the rights. There are probably a lot of species there, like for Samba and Capicola, that you find. You will find a lot of species. And that's the interesting thing, is that as it's really in the middle of the road, it will explain the road itself, like if it's out of Africa or in Africa. So we really need this species to better understand the connection between the African fauna and the Palearctic fauna.
Well, one interesting observation I'll make, this Meganomiine genus Euromonia was described from Kenya. But Pulowski from the California Academy collected it in Mali, and I've got a specimen from Mali, and it looks like, you know, it's identical to the figures in Michina, so it's the same species. And some years ago, I had a student who was actually studying ants, and he did some species distribution modeling for the European fire ant. And his data showed that there is an area along the southern Sahel that joins Mali and Kenya. It's quite narrow, north-south, but it's very wide. And it has similar climatic conditions. And so my guess there is that you've got the possibility for east-west migration dispersal based upon that narrow habitat range. And apparently, you know, there are some spots in the southern Sahel which are really quite diverse and rich, but you know, it's not exactly the easiest or safest place to travel these days.
Yeah, for sure. I saw some specimens of Euromonia in the collection of Schwartz, also of Max Schwartz, from there. I don't remember if it was Mali or Niger, but it was from this just below the Sahara.
Yeah.
Yeah, for sure. This area and also up. I would love to go also in the mountain in the middle of Algeria. All this area, it's very interesting, but most of the time it's not really stable in the political point of view.
No, well, you're going to have difficulty collecting in Britain soon, the way things are going.
Yeah, but it's really crazy. You think that, for example, now the trip that I did in the Baikal Lake could not be possible.
No.
So it's like, because Europeans cannot travel in Russia now, and so it's really a pity. It's I hope that we're gonna be better in 10 years, but it's not going better now.
No. Tadauchi's done a lot of collecting in Central Asia, Kazakhstan in particular. Osamu Tadauchi in Japan.
Okay.
He might be worth getting in touch with.
Yeah, I mean, Central Asia is still okay. Uzbekistan, Tajikistan is still okay to go. So we're gonna try to go next year with the team.
Oh, that's gonna be fabulous. I'm so envious. All right, any more questions? We've reached the end of the questions that have been sent in the Q&A. Lots of people are saying thanks for a great talk in the chat, and I will reiterate that. That was a lot of fun. It was great to have a survey of the group of beads that is sister to all of the others. Lots of really interesting insights into that. So thank you very much.
You're welcome. Thank you.
Thank you. Right, bye for now.
All right.
And the next talk is in a month's time, and it's going to be by Klaus Rasmussen. Do we have a title from him yet, Victoria?
A very broad bees from north to south. So I'm not entirely sure what that's going to encompass, but I'm sure
bees from north to south. All right.
Well,
I look
forward in so many
to
different
that.
areas that's what he's doing.
Yeah, probably from South America to Denmark, I would say. These are the two places of close, yes.
Well, he doesn't get many stingless bees in Greenland.
No.
All right. Well, thanks a lot. That was fabulous.
And just a reminder,
so much.
Sign up for the next talk and any future ones at yorku.ca/bees/packer. Great. Thank you, everyone.
Thank you. Take care.
Yeah, and you. Bye.
Bye.